What's up, guys? I'm here today to talk to you about semester 2 EOC exam review for biology. We're going to be getting into Mendelian genetics for our review section. Let's see what we've got here. We are currently on page 22 of the review, instructional sequence 1 from unit 3. This is stuff from in January, once we came back. Let's see what we've got here. Do I have good text boxes? I do. Let me get ready to add these in. All right, we start out with our first question here. What is the difference between homozygous and heterozygous genotypes? Well, let's reference a couple things here real quick. First, we will make note of the fact that genotypes is referring to the two-letter code that represents which alleles we have. For a certain trait. There's a couple ways these can be shown. We've talked about dominant and recessive in the past. Dominant is going to be capital letters. And recessive will be lowercase. If we are talking about something that is going to be heterozygous, heterozygous is going to be different. So for example, we could have a genotype that looked like something like capital A, lowercase a, or big B, little b, or big R, lowercase r. If we're talking about something that is homozygous, we are referring to something that would be like big A, or let's see, little f, or big Y, or even little u. The key for homozygous is homo tells us same. So we're going to have two of the same type of letters representing the alleles that we have for this. Hetero tells us different or other, and that's where we will be seeing one big and one little for those. Question 2 here. In cats, short hair is dominant over long hair. What phenotypic ratio and genotypic ratio would result from a cross between two heterozygous cats? Okay, let's make sense of this here. They're telling us that short hair is dominant over long hair. That means when I see things that have capital letters, that's going to be represented with our short hair. S's tend to be difficult to deal with because big S and little s don't look all that different. L's we can tell the difference between, but since long is recessive, that doesn't work. So we're going to use H for hair as we show this. And we're going to set up our little Punnett square here. I understand that on your paper, you may not have the space to do this the way that I am, but again, fit things in however you want. You can still utilize scratch paper or whatever else if you needed to. So we know that we are crossing two heterozygous short hair cats. That means for our H's for hair, I'm going to have a big H, little h for one parent, a big H, little h for our other parent, and we're going to do our Punnett square. Keep in mind that when we do Punnett squares, our letters that are on the top are going to drop down through both boxes, and our letters on the side are going to come across through both boxes. So if we fill this out, we'll bring my big H across and my little h across, and then my big H down, and my little h down and down. I can see from this that I have three of them that each have I've got three of them that each have at least one capital allele. That tells me that I'm going to have three that are going to have short hair and one that's going to have long hair. My one with the long hair will be this homozygous recessive one here. So that's going to give me a 3-to-1 ratio for my phenotype, what these guys are actually going to look like. However, when I do my genotypes, I have to break this down a little bit farther. Erasing this sucks. Okay. When I go to this here, I can see I have one that is homozygous dominant with big, big. I have two that are represented heterozygous style, and I have one that's going to be shown as homozygous recessive. I'd end up with a 1-to-1 ratio as far as my genotypes go, but I'd end up with a 3-to-1 ratio as far as my phenotypes go. That difference between genotype and phenotype is going to be big. They might not ask you that specifically on a test, like what's genotype or phenotype, but it'll get used in a bunch of questions. And so knowing what those are will be an important thing for us. Question 3. What approach would you use when examining the inheritance of two traits? Back in the past, we would use something called a dihybrid cross. But now we don't really do that anymore. This would have been the big 16-box Punnett square. The approach we're actually going to use in reference to this is going to be slightly different. And what we're actually going to see here is you're going to do two separate Punnett squares, then multiply the probabilities. I know I misspelled that. I've misspelled this quite a bit. Multiply the probabilities. So if I'm going to be doing this and I find out when I do my Punnett squares that one of them, darker color here, if I find out that one of them is a 1 out of 4 chance and one is a 3 out of 4 chance, I'm going to multiply these together. Multiply my top numbers for a 3, multiply my bottom numbers, 4 times 4 for a 16. I would have a 3 out of 16 chance of getting that specific combination if my two probabilities were these from my two separate Punnett squares. So instead of doing a massive 16-box Punnett square and use 4 letters in each box where one little letter can make a huge mistake and ruin everything for you, we now do 2 separate Punnett squares and multiply their fractions together to give us the probability of getting both of those traits in one organism. Question 4. Freckles are recessive. Unattached earlobes are dominant to attached earlobes. What are the possible outcomes for a freckle-attached earlobe woman and a homozygous non-freckled unattached man? Wow. All right, let's get this going here. So our woman has freckles, which are recessive, and attached earlobes, which are also recessive. That automatically tells us what we're working with here. I'm going to do her 2 Punnett squares and her 2 probabilities to get that one common fraction here. So back to annotations. The office is not calling us right now. So if I were to set this up for Freckles first, I know that she is recessive. So she's going to be bringing little f for Freckles. On my Punnett square that has to do with earlobes, she's also recessive because she has attached earlobes. So she's going to represent little and little. And if we have a man who is homozygous for both of these, that's going to put him as big F, little f, and big E, little e. And now we run our 2 Punnett squares. We can find the possible outcomes by looking at the way this plays out. If we drop these through, I can see here, I've got a 1 out of 2 or a 50% chance of having freckles or not having freckles. And on our other, I've got roughly the same, a 1 out of 2 situation for having attached earlobes or disattached earlobes. Now, if they wanted a specific combination, hey, if these were the parents, what are the chances of their kid having freckles and unattached earlobes? Well, that would be 1 out of 2 from the first one, 1 out of 2 from the second. I would multiply these together, giving me a 1 out of 4 chance or a 25% chance to have both of those specific traits. If I ended up with a different set of parents and these fractions were different, that would obviously lead to a different outcome here when we went to figure out the chances of their kid ending up a certain way versus another. Describe the non-Mendelian genetic traits. Let's see what we've got here. We've got a little matching section. Let's see what we can do. Controlled by genes located on the X chromosome. This one for sure is going to be D. Anytime we're talking about the X chromosome, we're referring to sex-linked. This is again because if you are female, you would have 2 X chromosomes as your sex chromosomes. And if you were male, you would have an X and a Y. The fact that a guy only has 1 X means that he would only be receiving 1 allele, like big B, and then nothing because there's not a second X, whereas a female would end up getting 2 copies for whatever trait it would be if it was found on that X chromosome. So that's going to lead to a different outcome depending on if you're male or female, and that again has to do with how many X chromosomes you have on your 23rd pair. So anything with things being specifically on the X chromosome is going to be a sex-linked trait. Base a little bit here, and we are back. Both alleles expressed in a heterozygous phenotype. Okay, both of them are expressed. That means that they are both showing up all the way. This is something that we're going to call codominance. If you were co-workers, you both work at the same place. If you were co-captains, you are both captains of the same team. Now, the fact that they're telling us that both are expressed and it's heterozygous tells me that more than likely I ended up with something like this. 2 dominant alleles somehow that code for different things. We call this one codominance because if they're both showing up, they're both fully dominant. So they're going to be dominant together at the same time. We call that codominance. The next one, many genes cause a blend of traits. If we're talking about a blending of traits, we are talking about polygenic. Polygenic literally means many genes. Some examples of polygenic stuff would be like skin color or your height. You have 3 separate genes and 2 alleles for each of those genes. So 6 of our alleles working together to decide how tall you're going to be or what your skin tone is going to be. So E for polygenic here. More than 2 alleles for a trait exists. Well, if there's usually, are you like this or like that? Do you have attached earlobes or no attached earlobes? Do you have freckles or no freckles? That's the standard type of breakdown for genetics. But if we have a situation where there's more than 2 options, we call this F multiple allele. At least they gave it a name that literally means exactly what we're talking about. Multiple allele is going to be things like blood type, where you could be type A, type B, type O, type AB. There's more options that are available for those. We call that multiple allele. And our last one here, heterozygous phenotype, is an intermediary between 2 homozygous phenotypes. This is something that we're going to call incomplete dominance. With incomplete dominance, we're talking that neither of those two phenotypes actually shows up all the way, you're going to get an intermediate form or a mixed form of it. This is going to be something like red flower, white flower. We breed those two together, and their kids that end up with a big R and a big W end up pink as that intermediate form because they had some of their stuff making the red pigment and some making white, which leads to a mixing. So incomplete dominance, neither of them shows up all the way. So we get pink, which is the middle form, versus codominance, where they both show up and we end up seeing both versions of that trait, such as like a white flower with red spots. That would be more of a codominance type thing. We clear our drawings. I lose annotations to scroll. Oh, a pedigree. Label the pedigree to identify inheritance and disease patterns. Some things to remember about pedigrees. First thing we want to recall about pedigrees. Circles are female. Squares are male. Shaded means has the trait. Unshaded does not have the traits. If it is half shaded, which we don't see here, but half shaded doesn't have the traits, but carries a gene for it. Now, as we look at this here, one more thing to keep in mind, which I'm not actually going to type onto here, but I am going to draw onto the screen. If we have a situation where two shapes are connected from side to side, meaning that the line enters their shape from the sides, this is indicating a marriage. If we have a fork that comes down, grab a different color. If we have a fork that comes down and enters the shape through the top, we are talking about a child relationship, like kids to parents kind of a thing. So if we look at this one here, the first thing I want to try to identify is this dominant or recessive? So I'm going to look at these parents here and I'm going to make some guesses. If I'm talking about dominant and recessive, let's say that this one here, let's guess that it's recessive. That would make this person big A and then something. We don't know what their second letter is. Actually, we do. It has to be little a. So I look at this and I go, okay, could this sick kid right here, could this sick kid right here get two little A's? Well, we get one little A from dad here, one little A from mom. That's possible. Would this kid be able to get a dominant A? Yep, from mom. Yes, again from mom. And we would be able to get one little A from dad and one little A from mom to have this son over here be sick as well. I look at these other two parents and I'd look at this and I'd go, yeah, I actually would guess that this is recessive because I can see these outcomes. I'm going to show you a non-example here. Keep in mind that this is incorrect, but I'm showing you this on purpose to try to tell what the difference is going to be here. If we say that this trait is dominant, actually, this would work with dominant as well. So I guess that example doesn't really make the most sense. But the keys for reading this, the way to tell if it's dominant or recessive, I am going to type this part on the screen because this part is useful. All right. If you're trying to decide whether or not something is dominant or recessive, what you want to look for is, are there any kids whose parents are not affected, but the kids are affected? What this is going to do is create a situation where if I had my parents as heterozygous, like big A, little a, I would be able to have a kid who had the trait and was recessive. If both parents were big A, little a, I'd be able to take a little a from both parents and have an affected kid. But you cannot have a recessive trait where the parents are affected, but the kids are not. If we had a situation where both parents were little a, and then we would have a kid underneath somewhere that was supposedly like this, big A, little a. If we had this type of a thing, this would not play out the way we think because no parent would actually have that dominant A to give the kid. So the only way you can have a kid that's different than the parents is if the trait is recessive. Recessive trait can have a kid that's different than the parents, but a dominant trait could not have kids. This is a terrible example that they put in here. I used to be all about this review. I'm not happy about this part. All right, anyways, let's move on here, guys. We've got what we need. We'll look at a little bit of the vocab here. Nothing too out of line. We've got a bunch of the major words here. So alleles again, each version of a trait. Genotype, which two-letter code represents your trait. Phenotype, what you look like based on the code. Dominant, capital letters, covers recessive, if heterozygous. Recessive, lowercase letters, gets covered if heterozygous. Punnett square, you guys know those are our 4-box windows that we use to test things. Heterozygous, different alleles, like big A, little a. And homozygous, two of the same allele form, like big B, or little u, or capital P, kind of a thing. We're going to start a new video for this next one, so I will catch you in the next one for evolution.